Hello friend, kindly subscribe to this channel DH Media GH, like this video, drop a comment and enjoy your studies. May God bless your efforts. Great. Ladies and gentlemen, this is another question and answer session with Daniel Clifford. Today, the aim is to attempt the question, what techniques are employed in studying medical arthropodology? Studying medical arthropodology encompasses various scientific techniques for understanding the biology, behavior, and control of arthropods involved in disease transmission. These techniques are vital for developing strategies to prevent and manage vector-borne diseases. Key methods employed in this field include 1. Morphological and taxonomic identification techniques. Understanding the morphology, that is, the shape and structure or form of arthropods of medical importance and their taxonomy is fundamental to identifying disease vectors. In medical arthropodology, we use microscopes to examine the physical characteristics of arthropods to identify and classify vector species. Such characteristics or traits include their body parts, such as antennae, legs, and wings. Taxonomic keys are guides used to classify and identify arthropods based on their physical traits. And dissection is a method used to examine the internal structures of arthropods to aid in species identification and classification. The internal structures include reproductive organs and digestive systems. 2. Molecular techniques. Molecular techniques are employed to identify arthropods and pathogens carried by arthropods. These techniques are useful in studying their genetics and resistance to control methods. In medical arthropodology, polymerase chain reaction, or PCR, is used to identify and detect the presence of specific pathogens. For example, malaria parasites and dengue virus within arthropods, even at low concentrations. DNA barcoding is a method that uses short genetic sequences to identify species of arthropods and distinguish them from other organisms. Gene sequencing is a technique that helps to study genetic variations in arthropod populations. It provides insight into resistance to insecticides or pathogens. And next generation sequencing, or NGS, is a technique that allows the study of complex microbial communities within arthropods, such as viruses and bacteria, which are critical for disease transmission. 3. Food sampling and surveillance. Food studies and surveillance are essential for monitoring the distribution and behavior of arthropod vectors in specific areas, particularly in regions prone to vector-borne diseases. The three key techniques are trap collection, larval collection, and outdoor surveillance. Trap collection is a strategy in which different types of traps, for example, light traps, carbon dioxide traps, and gravity traps, are used to capture flying insects such as mosquitoes in the wild. Larval collection is a strategy that involves collecting larval stages of arthropods, especially mosquitoes, from water bodies to help assess breeding sites and samples for experiments. And the outdoor surveillance is a technique that involves monitoring insect populations in the field over time, often through regular site visits to track changes in abundance, species distribution, and infection rates. 4. Behavioral studies. Studying the behavior of arthropod vectors is important for understanding how they interact with their environment and hosts. This helps to develop better control methods. The techniques involved relate to feeding preferences, flight and dispersal patterns, and attraction studies. About feeding preferences, note that research into which hosts arthropods prefer to feed on, for example, human versus animal, can help in designing control strategies like repellent or bird nets. Research into which hosts arthropods prefer to feed on, for example, human versus animal, can help in designing control strategies like repellent or bird nets. The flight and dispersal patterns are about studying how far and how often arthropods move in search of food or breeding sites. And attraction studies techniques enable researchers to evaluate how arthropods are attracted to environmental factors such as light, heat, or chemical cues such as carbon dioxide or human sweat. Such evaluation is useful for designing traps and repellents. Number 5. Other pathogen detection and analysis. A critical part of medical anthropology includes detecting and studying the pathogens that arthropods transmit. In addition to the molecular techniques used in detecting pathogens, enzyme-linked immunosorbent assay or ELISA, immunofluorescence, and virus isolation are also used. Enzyme-linked immunosorbent assay, or ELISA, is a technique used to detect the presence of specific antibodies or antigens in arthropods. It helps identify diseases like dengue or malaria. Immunofluorescence is a method of using antibodies tagged with fluorescent markers to detect pathogens within arthropods under a microscope. The virus isolation method involves growing viruses from arthropod samples to study their properties and transmission dynamics. Insecticide-resistant testing. Number 6, insecticide-resistant testing. Insecticide resistance is a major challenge in vector control operations. Various techniques are used to evaluate how arthropods respond to chemical treatment. Techniques involved in insecticide resistance testing include biological assays or bioassays, synergy studies, and genetic tools or procedures for analyzing resistance in vector species. Bioassays involve testing the mortality rate of arthropods exposed to insecticides to determine the effectiveness of different chemicals. Synergy studies involve investigating how combining insecticides or using different formulations can overcome resistance. And genetic analysis of resistance involves studying the genetic mutation or mutations in arthropods that lead to resistance against insecticides. Data generated through these techniques help in developing new control strategies. Number 7, mathematical modeling and GIS or Geographic Information Systems. Mathematical models and GIS are used to predict the spread of arthropod-borne diseases and assess vector control strategies. Techniques include mathematical modeling and GIS mapping. Mathematical modeling is used to understand the dynamics of vector populations and disease transmission, including factors like climate change, human behavior, and control measures. And GIS mapping combines field data with geographic mapping to track the distribution of arthropod vectors and assess areas at high risk of disease transmission. Number 8, laboratory culturing and rearing. Laboratory-based methods are used to breed and study arthropods in controlled experiments or environments. This is essential for experiments on their life cycles, vector-host interactions, and responses to control measures. The techniques are related to rearing mosquitoes, artificial feeding systems, and insect colony maintenance. Regarding the rearing of mosquitoes, note that mosquitoes are bred in the lab to study their behavior, life cycle, and interaction with pathogens. The artificial feeding systems are used to stimulate, I beg your pardon. The artificial feeding systems are used to simulate animal or human blood meals for studying feeding behavior and pathogen transmission. And the insect colony maintenance involves rearing and sustaining colonies of specific arthropod species to conduct long-term studies on their biology and susceptibility to treatment. And lastly, number 9, vector control techniques. Developing and testing new methods for controlling arthropod populations are two critical focuses of medical arthropodology. Such vector control techniques include the release of sterile insects, genetic modification, and biological control techniques. The release of sterile insects involve releasing sterilized male insects into the wild to reduce the population by preventing reproduction. The release of sterile insects is a technique which involves releasing sterilized male insects into the wild to reduce the population by preventing reproduction. The genetic modification is about creating genetically modified arthropods, such as mosquitoes, that are resistant to pathogens or unable to reproduce to reduce the vector population. And the biological control involves introducing natural predators, competitors, or parasites to control arthropod populations. For example, introducing fish to control mosquito larvae. In conclusion, note that the techniques employed in medical arthropodology are diverse and interdisciplinary, ranging from morphological identification and molecular analysis to field surveillance and behavioral studies. These techniques allow researchers to understand the biology of arthropods, their role in disease transmission, and to develop effective control strategies. This is Daniel Hifford. See you in another question and answer session. Have a nice day.